So we will now move to panel session 3. The topic now is the impact of modern lifestyles on the human microbiota. We again have a multidisciplinary panel that is learning and has learned in many cases to speak the same language, and that's true here. We have a wonderful moderator who again comes from the world of ecology, also engineering, and you will see that the built environment, the modern built environment, is a major issue amongst many for this session. We also have a wonderful immunologist and a wonderful microbiologist to deal with the role of the immune system and diet in accommodations of modern lifestyle impact. So I want to now introduce Jessica Green, who will moderate this session. Jessica is the Alec and Kay Keith Professor in Biology at the University of Oregon and professor at the Santa Fe Institute in New Mexico. She's also co-founder and director of the Biology and Built Environment Center at the University of Oregon, which she helped to inspire and build and lead, that directly addresses some of the issues that you're going to hear about now. She has done some wonderful work in this area and thought very broadly about some of these large global issues, and it's a real delight to have Jessica here. Jessica.
Okay. So can you all hear me? No? Okay, so I'm on. Can you hear me now? No? Now can you hear me? Okay, great. It's our job to, it's my job at the beginning to wake you all up. So this session is focused on the impact of modern lifestyles on the human microbiota, and I'm going to introduce our diverse panelists here. We have Justin Sonnenberg on my left. He's an assistant professor in the Department of Microbiology and Immunology at the Stanford University of Medicine. He is the recipient of the NIH Director's New Innovator Award and also the Burroughs Wellcome Fund Investigators in Pathogenesis of Infectious Disease Award. The overarching focus of his lab at Stanford is on host microbe systems biology, with an emphasis on changes in the gut microbiota in humans over evolutionary timescales. To his left is Ruslan Mezatov. He's a Howard Hughes Medical Institute investigator, a David Wallace Professor of Immunobiology in the Department of Immunobiology at Yale University School of Medicine. He's one of you. He was elected last year into the Institute of Medicine, and he's also a member of the National Academy of Sciences. His research focuses on the biology of inflammation, innate immunity, mechanisms of allergic reactions, which we talked about at lunch today, cell signaling, and gene regulation. And we have remotely joining us Amanda Henry. I don't know if we'll have... Hello. Hi, Amanda. Amanda is an anthropologist, and she is the research group leader of plant foods and hominin dietary ecology at the Max Planck Institute for Evolutionary Anthropology. I was interested to learn that she was the first, she carried out the first research to show that Neanderthals consumed plant foods, and she did this by studying the plant remains in the dental calculus, or in teeth. And she has she's now focusing on research in the gut microbiome, looking at differences in non-industrialized versus modern societies. So I think this is a fantastic combination of expertise and perspectives, and what I'm hoping this group is going to challenge you all and one another to think about are the opportunities and also the challenges for connecting the really different approaches that each of the panelists bring to the table. Justin and Ruslan have both have experience using highly controlled experimental approach with model organisms like mice to understand how modern lifestyles might impact the gut microbiota, and Amanda operates in a much more complex world. So she thinks about human culture, human behavior, human practices, and how that might impact the gut microbiome. So I'm sure that by bringing these minds together, it'll foster great ideas for future research. What I want to do now is give you some idea of my perspective and my research program and how it relates to everybody that's here in this panel. I was trained as a nuclear engineer. My Ph.D. advisor, I'm thinking about him a lot today. He was inducted to the National Academy of Engineering while I was in his lab, and his expertise was in risk analysis. He really codified risk analysis in the nuclear industry, and I've been delighted to hear risk analysis brought up a lot today. David Relman talked about it this morning in the session. And when I first entered the field of ecology, it was from a theoretical perspective in trying to model and understand how biological communities assemble. And when I first learned about human microbiome research, one of the first questions that I asked is, where do our microbes come from? We know that we get microbes from the food that we eat. We know that we get our microbes from our mothers. We know that we get microbes from our friends. And something that I'm going to talk to you about now is the potential role of the built environment. Everyone in this room is going to spend more than 90% of their lives indoors. And given the amount of time that we spend indoors, it's very intuitive that we are exchanging microbes with the built environment. But what we don't understand right now is the relative contribution of this environment, this habitat of ours, the microbes that are in it and the human microbiome, relative to other contributions such as our diet and other lifestyle choices. I really got interested in this work because of the pioneering thinking by Paula Olszewski, who's here today. She's a program officer at the Alfred P. Sloan Foundation, and they've really invested a lot in driving research to understand the built environment microbiome, with the understanding that this would ultimately lead to understanding the interactions between the indoor microbiome and health. So here you're looking at a man who's sitting in the built environment. We know that he's exchanging some microbes with what is on the couch. Some of those microbes are also being contributing to his microbiome. And when I was thinking today when in some of the earlier talks about how great it would be when we look at the relationship between mothers and the microbiome of their children, if we could also include their fathers in those types of figures, because that might help us understand our children looking like their mothers in the early ages because they're in the same household, or is there something special about interacting closely with their mother? I wanted to tell you a little bit about how we think about the built environment microbiome. So microbes come into the built environment two ways. They come in with air and they come in with water. And the way that modern buildings are designed has fundamentally altered the kinds of microbes that are indoors. Buildings before the air conditioner was invented had natural ventilation and natural light. And now, many of us spend a lot of our times in rooms like this, where the air that comes into the room is treated, it's filtered to keep a lot of the bioaerosols outdoors. And it's also treated to be at a specific temperature and relative humidity. So the modern building has definitely impacted what kind of microbes are in the built environment. And we know that water is also treated in ways that it was not once. So this is impacting what's indoors and the types of microbes that we're interacting with. People and their pets and their plants and all of their stuff bring microbes indoors as well. One human being in the course of an hour contributes to indoor air over 40 million bacteria alone. This isn't including viruses, this isn't including fungi. So that's more bacteria than the number of people that live in the state of California. And they do this by resuspending microbes from the surfaces that they touch and also directly shedding their microbiome. We know that pets have an impact on the indoor microbiome and potentially human health. Susan Lynch did a great study last year where she looked at the dust in the homes of people who had pets and who didn't. And she found that if you expose mice to the dust with people who do have pets, it changes their gut microbiome and their immune function for the better. We know that environmental conditions, just like any habitat, impact the indoor microbiome. So right now, we humans live in what's called a thermal comfort zone, where we have a very narrow range of environmental conditions that we live in, which is very different from the natural variation that not very recently in evolutionary timescales we were exposed to. And I am regularly thinking about how this homogeneity is selecting for particular microbial types indoors and whether or not this matters for the human microbiome and human health. We know that the different types of materials that we bring into the built environment are going to select for different microbiomes. And there are a number of research groups that are now looking at how synthetic versus natural materials colonize different microbial groups. And finally, we know that we're disturbing the built environment on a regular basis by using antimicrobial products and antibiotics. And this is another very active area of research where we're trying to understand how does that impact the built environment and in turn human health. So I'm going to close by reminding us all that we spend 90% of our lives indoors. Every breath that you take, you're breathing in bioaerosols that are loaded with bacteria, fungi, archaea, and they're getting into your mouth, possibly into your gut. Your skin is exposed to microbes during this time. And we're still trying to assess, because we don't know the relative contribution of airborne microbes and surface microbes on fomites to the human microbiome. Of course, it's going to vary depending on if you're interested in the skin or the gut or the mouth, but the interaction between these two microbial ecosystems is of interest. And the reason for the overarching motivation for thinking about this is because right now, architects are unintentionally designing the indoor microbiome. And if we learn about how building design and operations impacts indoor microbiology, there's a possibility that we could positively impact human health. So with that, I'm going to turn the conversation to Justin.
Great.